Okay, so due to Rosalind Franklin's work, as well as Watson and Crick, James Watson and Francis Crick, what they did is they took her work and some work that they had been doing, and they came up with a model to show what the shape of DNA is. And they are the ones that discovered that DNA is a double helix model, similar to a twisted ladder, that the sides of the ladder are made up of sugars and phosphates, and that the nucleotide bases, the A's and the T's and the C's and the G's, make up the steps or the rungs of the ladder. Again, you can see that complementary base pairing is mentioned here, that always a pyrimidine and a purine are bonded together. The A is a purine and the T is a pyrimidine, and the G is a purine and the C is a pyrimidine. And then in 1962, Watson and Crick received the Nobel Prize. Rosalind Franklin was not part of that because she had passed away. So they don't give the Nobel Prize to people who are, no longer living. This shows you the double helix structure of DNA, and it shows you the sides of the ladder are sugars and phosphates. And if you just look at picture C, you can really see that. This little yellow circle with a P, that represents a phosphate group. And then the pentagon shape with an S, that's the sugar. So each nucleotide has a phosphate, a sugar, and then a base. This base is a guanine, and then phosphate, sugar, thymine. And over on the other side, and it's oriented with the P's kind of facing the other end, you have phosphate, sugar, adenine, phosphate, sugar, cytosine. So you can see that always a pyrimidine is paired with a purine, so that the steps of the ladder are always going to be a total of 3 rings, so that the steps are always the same width. Okay, now we're on to section 2. Just a review of section 1, you need to make sure that you understand that the scientists that played a role in discovering that DNA is the genetic material, those scientists are Frederick Griffith, who discovered transformation, Oswald Avery, who confirmed the transformation, that DNA is the transforming molecule, and then Hershey and Chase, who did their experiments on the bacteriophage, the T phage, and discovered again that DNA is the molecule that contains the code to make new viruses. So those are all the scientists involved in discovering that DNA is the genetic material. And then we had the scientists that were involved in the structure of DNA. That would be Franklin, Watson, and Crick. And remember Chargaff, who learned the complementary base pairing, discovered complementary base pairing. So that's a history lesson. And now we move on to DNA replication, which is the process of copying a DNA molecule. DNA replication is semi-conservative. In other words, each new DNA strand has one old strand and one new strand. So for every molecule of DNA, if we look at this original parent strand here at the top in picture B, one side or one strand is from the old DNA, and then the other side or the other strand is new. So if this is the parent strand up here at the top, what it does is it splits, and one strand from the parent strand is used as a template to create a new strand. So the nucleotides for the new strand are added on to the old strand because of complementary base pairing. So each strand of DNA has one old and one new strand. That's semi-conservative replication. All right, so how does this process work? Well, first we have to have separation of the 2 original strands, and that occurs through an enzyme called DNA helicase. And DNA helicase unwinds and separates the old strands of DNA. And then we have single-stranded binding proteins, and what they do is they attach to the newly separated DNA to prevent the helix, the double helix, from like coming back together and reforming. So we have DNA helicase that separates the two strands, and then single-stranded binding proteins that basically holds them open. Then we have what the complementary base pairing that adds new nucleotides to the template DNA strands. This begins with the addition of a primer on the strands to be replicated. And the enzyme that adds the primer is called DNA primase. So DNA primase places a primer on the strands that are about to be copied. And then DNA polymerase is the enzyme that comes along, recognizes the RNA primer. That primer is a short piece of RNA. And recognizes the primer and begins adding nucleotides that are complementary to the old strand. Okay? And it's easier to explain this when you're looking at a picture. So looking at this picture with one of the template strands, this big brown molecule is DNA polymerase. DNA polymerase begins at one end and starts adding nucleotides continuously down that strand. With the other strand, the DNA polymerase adds little pieces of DNA. So you end up with these fragments. You end up with a little bit is replicated, a little bit is replicated, a little bit is replicated. And these fragments of new DNA are called Okazaki fragments. So when all of that DNA has been replicated, those fragments still have to be joined, and they are joined by DNA ligase. This is a process really that if you go back to the textbook, to the e-text, and watch the video, the animation tutorial of DNA replication, that's very, very helpful. So let's start with the whole process. First, you have your parental DNA double helix right here. The first thing that has to happen is the enzyme DNA helicase has to unwind and unzip and separate those strands. And then you have the addition of single-stranded binding proteins, these little, I guess they're orange, little molecules here, and they're going to keep the strands open. And then you have the addition of an RNA primer by DNA primase, and that's going to be where the DNA polymerase will recognize and begin adding nucleotides to the template strand. So DNA polymerase is going to add nucleotides that are complementary to the template strand. And on one of the strands, this will be done continuously from one end to the other. And the other strand, which is called the lagging strand, the leading strand is replicated continuously, but the lagging strand is replicated in pieces. So you have the little purple primer here, and then you have DNA polymerase adding a few nucleotides and forming an Okazaki fragment. Then you have another Okazaki fragment, another one. So at the end of the replication, DNA ligase will seal the Okazaki fragments together, so there'll be one continuous DNA strand. There's a section on prokaryotic DNA replication versus eukaryotic. We're going to skip that because it's just a lot of information, so we're trying to shorten it a little bit. So there is a little section on telomeres. Telomeres are the ends of chromosomes. And DNA polymerase is not able to replicate the ends of the chromosomes, or telomeres. So telomeres are copied by telomerase, an enzyme called telomerase. And sometimes when this telomerase activity is unregulated, it can cause problems with cell function. Telomeres have found to be important in aging, and there's a lot of research on telomeres and the enzyme telomerase. But the ends of chromosomes are replicated by a different enzyme than DNA polymerase. Okay, so accuracy of replication. You can imagine that if we're talking about 150 million base pairs in our entire genome, that there's going to be some mistakes made. That sometimes instead of adding an adenine, that DNA polymerase is going to add a cytosine, So DNA polymerase is actually really, really accurate, but it makes a mistake about once in every 100,000 base pairs. But DNA polymerase is also capable of identifying and correcting the mistakes that it makes. So for the most part, when it does make a mistake, it corrects that mistake. Okay, section 3 is about RNA. And we're going to skip down to, okay, so the one gene, one enzyme hypothesis is a big hypothesis, or it's been developed into a theory. It's the hypothesis that one gene or piece of DNA codes for a specific protein. And we know that the flow of genetic information in a cell starts with DNA, and then that DNA inside the nucleus undergoes a process called transcription and makes a molecule called messenger RNA, which is complementary to one of the strands of the DNA, the template strand. Okay? So if you look at this template strand, for example, the first nucleotide base is a T. So the first one in the messenger RNA is an A. The second one is a C, so then you have a G. The third nucleotide in DNA is a G, so you're going to have a C. But the point is that the messenger RNA is complementary to the template strand of DNA. That messenger RNA is a smaller molecule, so it's able to leave the pores in the nucleus and go into the cytoplasm, find a ribosome, and attach to it. And remember that we learned that ribosomes synthesize proteins, and this is how they do that. The messenger RNA has a code that codes for specific amino acids. So here's an example. Every 3 nucleotide bases in messenger RNA is called a codon. So every 3 bases is going to code for a specific amino acid. A, G, C is going to code for an amino acid called serine. GAC is going to code for aspartic acid or aspartate. And CCC codes for proline. So it is that code that leads to a chain of amino acids that we call a protein. And this is how DNA codes for our traits. It's DNA codes for RNA, and RNA codes for protein. So we need to talk about RNA a little bit. There's some differences. RNA is single-stranded, for one thing, instead of double-stranded. The nucleotides in RNA contain the sugar ribose instead of deoxyribose. So these are differences between RNA and DNA. RNA has ribose, DNA has deoxyribose. RNA has 4 nucleotides, and 3 of them are the same as DNA. They're A, C, and G. But RNA does not have the T. Instead of T, it has U, which stands for uracil. So RNA contains the nucleotide uracil instead of thymine. Now, there are 3 types of RNA. There's messenger RNA that we just talked about. Messenger RNA takes the message from DNA to the ribosome, and it also contains the code for the amino acids in the protein. Transfer RNA transfers, so each transfer RNA is only